All right, so a few things, loose ends, really quick. One of your classmates approached me after a lecture on Thursday and wanted to confirm about the epithelium in the large intestine, and I reiterated that the large intestine does not have any villi, but it is lined by a simple columnar epithelium, all right? And why is that? Because there's a lot of absorptive function going on there, right? So you have to have a means for absorption, but you don't have folds and you don't have villi, all right? Can we expect that the simple columnar epithelium has microvilli? Sure, all right, because there's a lot of absorption going on there, all right? So when you look at this figure here, we're talking about this epithelium running along the surface here, and then of course you can see there's going to be then a simple columnar epithelium there. There's going to be goblet cells there, of course, and more of them as you get further down the colon, right? Because you're removing liquid, it's becoming more and more solid, and the more that the undigested waste becomes solid instead of liquefied, the more you need mucus in there to enable their movement, all right? In addition, as you can see here, you've got some pretty robust intestinal glands, which are primarily for the production of mucus. So they are jam-packed with goblet cells, all right, and secreting up through the duct, as you see there, secreting up through their duct and providing a lot of mucus. And that's in the mucosa, right? We're in the mucosa layer of the gut there, all right? So nice, rich abundance of glands, and of course you see them, the openings of the intestinal glands there on the luminal surface, right? Okay. All right. So worth a second look, and looking at the actual micrograph here, you can see the coloration here of the intestinal glands, and you can see what do you notice in the coloration of these glands? Very light, which tells you these are mucus-secreting glands. All right, these are mucus-secreting glands. These are your goblet cells. Okay, all right. So that's one. Two. We looked at the pancreas. I'm not going to say anything more about it except to remind you of a basic vocabulary, and that is, we talk about the accessory glands of digestion, right? We've used that term back when we introduced the digestive system, the gross anatomy, and I want to just go back to it and reiterate that vocabulary. Accessory glands of digestion are what? Name them for me. Pancreas, liver, gallbladder, and salivary glands. Accessory glands. All right. And so those are the accessories, meaning that they are derived as outpocketings, right? The liver is it develops as an outpocketing of the gut, all right? The pancreas develops as an outpocketing of the gut, right? Because these are glands, and you remember how glands form, right? They're an outpocketing of the epithelium, all right? And eventually they grow into something quite robust in both these instances, all right? But they are apart from the main tube, right, from the digestive tract itself, the esophagus, stomach, small, large intestine, all right, they're attachments to it, and likewise the salivary glands, all right. Remember then also that we have then all these minor glands, then, like we were just looking at the intestinal glands, the things that are built into the mucosa, right, and whether we're talking about the intestinal glands or the Brunner's glands in the small intestine, I'm working in reverse order here, or the gastric glands in the stomach, all right, all in the mucosa layer. Those are all minor glands. And even in your mouth, right? In your mouth you have your 3 pairs of salivary glands, but you also have minor salivary glands built into the wall of your cheeks, all right? And so it isn't just these glands, okay? All right, but there's the minor ones as well. All right, so now let's take it back to where we left off last time, which was looking at the liver. And I've put just a sketch here. Obviously, it's not perfectly anatomical. We've sort of spread things out so that we could distinguish certain components here. All right. What I want to focus on, as we did last time in our conversation, is the relationship between the liver and the digestive tract. All right. And we were speaking about two particular components: the vascular component, which includes this vessel here, all right, carrying all the absorbed nutrients, right? Everything that's absorbed from the gut, from the stomach, from the small intestine, from the large intestine, everything that's absorbed from the gut eventually converges on the hepatic portal vein, right? The hepatic portal vein. It is the drainage, all right? But what is a portal vein by definition? It's a vein that begins with capillaries and ends with capillaries. It's a bridge between capillary beds, all right? And in this instance, you have the capillary beds, the capillaries that run into the villi that we looked at, where absorption is occurring, all right? And then they all converge to form this portal vein, all right, which is delivering about 80% of the blood volume going into the liver. Now, because it has already, it was at one time blood that came from the superior mesenteric artery to supply the gut, that blood has given up its oxygen, hasn't it? All right? It's given up its oxygen, which was used by the intestinal tract, all right? But it has collected all these nutrients. So it is venous blood, right? It's low in oxygen, but it's rich in everything that it's absorbed during the, what we call the absorptive state, which is when you're, from the time you eat a meal to the time it's fully digested, all right? And so that's all being transferred to the liver, but the liver also needs some oxygen, so we have the hepatic artery here making up the balance, the other 20%, all right, of blood going into the organ. And don't get too picky on these percentages. I was looking at your textbook today, and it says 75% or something, or 65, whatever. 75% is what it says. Don't worry about that. 80-20 is fine, all right? When this blood goes in here, then it's going to be processed by the cells that are in the liver, all right? And those cells are primarily hepatocytes, all right? And we're going to look at these, and we're going to also look at a group of cells called Kupfer cells, all right, which are our old friends, fixed macrophages, all right? They're fixed macrophages, and yes, they're monocyte derived. They're part of what's called the reticuloendothelial system. We'll come back to that, all right? So these two cell types, and what other cell types are you going to expect in here? Hint, vascular, of a vascular nature. You're going to expect endothelial cells forming the capillaries, right? That are carrying all this blood coming into the organ via the hepatic artery and the hepatic portal vein, right? And I mentioned that. You knew that, right? Endothelial cells for the capillaries that are perfusing the liver, all right? We're going to see that the arrangement of these endothelial cells is different from anything we've seen before, all right? We saw continuous capillaries where the capillaries, for example, that run through muscle tissue, all right, have a nice, robust endothelial wall. And then I mentioned to you another time that the capillaries that run through endocrine glands have little windows. Remember, I called them fenestrated capillaries. And that allows the product from the endocrine gland to gain access to the blood circulation, all right? So we can say that a continuous capillary is sort of the most secure kind of capillary. Stuff can't leak out that easily. A fenestrated, for good reasons, is kind of leaky, isn't it? It has those little windows. It's like taking a piece of Swiss cheese and rolling it into a cylinder. It's got lots of holes in it, all right? The endothelial cells here in the liver form what are called sinusoidal capillaries, all right? Sinusoidal. And these are even leakier, all right? Sinusoidal capillaries. All right, again, they're endothelial cells, but they have huge gaps between them. All right, and what does that mean about the ability of the hepatocytes to process what's in the blood? What does that mean about the amount of contact between what's entering into the liver in terms of the components of the blood and the working cells in the liver, the hepatocytes? It's huge, right? It's huge because you have these capillaries that are super leaky, and all kinds of stuff that's being conveyed in those capillaries then is going to be able to interact with the hepatocytes in here and with the Kupfer cells, all right, in the liver as well. And once we finish that processing then, all right, it drains through the hepatic vein, all right, and this is the inferior vena cava going back to the heart, carrying blood back to the heart. All right, and once it goes to the heart, it gets pumped out to the rest of the body again, right? So, as we've said before, everything that's absorbed here gets checked, processed, regulated by the liver, and then it goes on hepatic vein, inferior vena cava, to the heart, and from the heart out to every other part of the body, right? But the liver has had a chance to check it, right? So if there's alcohol in there, the liver's going to do its level best to pull the alcohol out before that goes on to the rest of the system. All right? If there's a lot of nutrients in there, the liver's going to take a share of them and save it for later and only allow some to go on to the rest of the body to feed you now. Right? And so on. If there's vitamins in there, let's store most of them here so we have a supply of vitamins for months ahead. All right? And we'll allow a small amount. We'll just dole out, as needed. Okay, so your liver is this checkpoint, all right, and its placement here demonstrates that. All right. Finally, we have this other function of the liver among many, and that is the liver is the source of bile production. All right, and bile, without going into too much of it, is a detergent. All right. In the sense that its purpose is to take the fat that's in the food you eat and emulsify it. All right? Now, that event is going to happen in the digestive tract, thankfully, because the bile produced by the liver, stored in the gallbladder, and ultimately traveling via the common bile duct is delivered here, meeting at the hepatopancreatic ampulla and entering in through the major duodenal papilla, right? And going into the small intestine. And so bile is an emulsifier. And to put that in really simple terms, by mixing the fat, which is in big droplets, all right, in your gut, with bile, it breaks it up into tiny droplets, all right? And what's the difference between big droplets and tiny droplets? The surface area of those droplets now is enormous, all right? The combined surface area. So what you're doing is you're taking a droplet of fat, okay, and you're breaking it up into many tiny droplets. And which do you think is going to be easier for digestive enzymes to work on? This or this? All right, which has a higher surface area? The collective here or this? All right, so I'll just for the record. This has a high surface area. This has a lower surface area. So this is what you want to do. And bile salts, all right, do that. All right. There's other stuff in the bile, by the way. All right. There's cellular waste products, specifically when your red blood cells wear out. You take hemoglobin, and you recycle the protein part of the hemoglobin. You recycle the iron component of hemoglobin, but there's another part of the molecule called the heme molecule, and it's a complex ring called a porphyrin ring. All right, you don't have to know that, but that can't be recycled. That has to be dumped. All right, and so your liver takes up the spent red blood cells, processes the protein that's inside them, the hemoglobin, takes that part of the hemoglobin and converts it into something called bilirubin. And bilirubin is known as a bile pigment, all right? And that is combined with the bile salts that are produced here. So that, too, is traveling in this common bile duct. And here you're dumping that into the digestive system. So that's kind of interesting, isn't it? You're dumping things into the digestive system, some of them because they're going to help you to digest food, and others because it's an avenue to get rid of those things, all right, through what's called fecal excretion, all right? And let me just resolve that word for you because it's not as special as you think. Okay. So the word, when you get rid of undigested wastes, food that you ate that couldn't be digested, that never entered your body, right? It stayed in the gut and it's going out the south end. That's called elimination, right? You're eliminating undigested wastes. That is not excretion, right? Excretion, by definition, is getting rid of cellular wastes, And so anytime you're adding something to the digestive tract that was actually breakdown products from things that were in the body, like bilirubin, a breakdown product of hemoglobin, that's going to be excreted, and it's being excreted with your feces, hence it is fecal excretion. Okay? Got it? All right, so we have then this duct here that is carrying... obviously, the liver is producing bile. That bile is going to be stored here in the gallbladder, where it's concentrated about 12 times. All right, and then through hormonal stimulation, you're going to pump those contents through the common bile duct into the digestive tract as needed. All right, and so we have then, as I finished up last time, we have a vascular system carrying blood into the liver and draining through the hepatic vein, and we have a biliary system draining away from the liver, right? So we've got this opposite direction, opposite flow here, don't we? All right, blood flowing in one direction, bile flowing in the other. We're all good? All right, so now we can dive in to the organ. And as you see up here real quickly, this is easy stuff. The liver is only partially partitioned, all right, incomplete lobes, and it has sort of 4 lobes if you want to break it down like that. All right, a left lobe that's pretty small because your stomach is over here, right? So the organ sort of makes it into that territory, but not very well. Over here you got lots of room. So the right lobe is bigger. All right. Now, the shape of the liver is kind of classic. You see it, you recognize it. But you know what? Its shape has nothing to do with its function, does it? It's just a bunch of cells dividing up here in the upper abdominal cavity and filling in the space where there isn't intestine, where there isn't spleen, where there isn't stomach. That's all. So the organ just sort of grows to fill in the vacancy that's there. And you know what it looks like when it does that? It looks like the liver, all right? That's the point. That shape up there is only because the cells are dividing and growing into filling the vacancy that's there, all right? And as it does that, part of those cells as they divide are encountering parts of the mesentery. And if you have mesentery that's in the way and the cells are dividing, guess what's going to happen? They're going to grow to either side of that, right? They're going to sort of straddle it and grow to either side. And so that membrane becomes sort of a semi-partition that's there only because the cells have grown onto either side. And so as a result of that, the liver gets partitioned into these 4 lobes. All right? The right and the left and the quadrate and the caudate. And is it important? No. All right? It isn't. It's all one big organ. It isn't like when we talk about the lungs, and we talked about the lobes in the lungs, and that was important. All right? Okay, so that being said, let's get into the microscopic anatomy real quickly. If I take a little tiny piece of liver, smaller than a millimeter square, all right, and I try to get down to the functional unit, to the smallest unit of the organ that I can, I'm going to see that the connective tissue within the organ, subdivides it into regions that look to some degree like hexagons. All right? I much prefer to be talking to a biology class who are interested in livers other than human liver, because the pig liver is perfect hexagons throughout. All right, the human liver, there's a hexagon here, there's a four-sided here, there's a pentagon over there, but you know what, we'll stick with hexagons. And what it is that we have these structures. Each is called a lobule. And there's our functional unit, a liver lobule. And the liver lobule consists of a bunch of hepatocytes, all right, that are arranged around a central vein, and they are arranged in chains, all right, of cells that are going out like the spokes of a wheel. All right? Good. These are all the hepatocytes. And the space in between these hepatocytes is what? That's where the blood flows. So the space between these hepatocytes is sinusoidal capillaries. You were going there, yes? All right, that's your sinusoidal capillaries. So now I've just shown you the uppermost layer of cells, and you have to now imagine many layers, or what we call plates of cells, all right, filling in this hexagonal cylinder, can I call it that? All right. Remember, it's microscopic. It's only a few millimeters at most.
Is that full top one hepatocyte, or is that many?
No. These are all hepatocytes. Each of these is a tiny little hepatocyte. All right. And this. All right. We'll get to that. All right. Now, coming up into each corner of the lobule, all right, there's going to be a branch of the hepatic portal vein. All right. Into each corner, a branch of the hepatic portal vein. And likewise, coming up into each corner is going to be a branch of the hepatic artery. Okay? And draining from each corner is going to be a tiny little, come on, give it to me, bile duct. All right? A tiny little bile duct. What do you have here? You've got exactly what we have here at the gross anatomical. All right? And what's going to happen then is the blood is going to drain in through these sinusoids, all right, inwards towards a drainage, which is the central vein. And guess where the central vein goes eventually? To the hepatic vein. There you go, all right? So there you see it. And, of course, this is a branch of the hepatic portal vein then going in, and this is a branch of the hepatic artery. So as I told you on Thursday, everything that happens gross anatomically is now happening at the microscopic anatomy level, at the level of each lobule, all right? This arrangement of hepatic portal vein, a tiny branch of hepatic portal vein, tiny branch of hepatic artery, and tiny branch of bile duct, together, this is called a portal triad. All right, and you get the triad part of it, the three elements then. All right, and so each corner has a portal triad. Okay, and blood is then draining in, hepatic portal vein blood, hepatic arterial blood, draining in and being processed as it moves from the periphery towards the central vein, being processed by all those chains of hepatocytes. Okay? Got it? All right. And all the different things that the liver does. Removing toxic materials, taking all the nutrients and deciding what you get to eat now and what gets stored as energy for later. Storing up vitamins. Removing those spent red blood cells, because the Kupfer cells that are sitting along intermittently down these sinusoidal capillaries, those Kupfer cells are gobbling up those spent red blood cells. Those old, 120-day-old red blood cells are being removed by the Kupfer cells, all right? And so you see a lot of functions going on here, all right? In addition, the hepatocytes are making your blood plasma proteins, all right, which is super important function, all right? So it is a gland that is secreting things into the blood as well as removing things from the portal from the blood that's coming through the hepatic portal vein. It's adding things. And what you're going to see, and I'm going to draw it over here on the side, is that if I have a chain of hepatocytes here and an adjacent chain of hepatocytes here, and we call this a sinusoidal capillary here. We won't draw the endothelial cells in it, but here's our sinusoidal capillary. If you can imagine another group of cells just below or to the side of this one, of this chain of cells. So far, so good? And if I were to cut in cross-section here and show you these two cells, here's what they'd look like. In between them, there's a little canal, all right? And that canal is separate from the sinusoidal capillary. This is a canal between neighboring cells, all right? And it is called a bile canaliculi. All right? And guess what's flowing there? The bile, all right, going in the opposite direction. So you've got blood flowing in this direction, and then if you'll permit me, then you've got bile flowing in that direction, all right? And that's going to drain into these bile ducts. Okay? There it is. All right? This whole thing is called a liver lobule. I think the one on the screen is much more elaborate and nicer than the one I've drawn, but you get the idea now. All right? You've got tens of thousands of these lobules in the liver. All right? And this is the little functional unit. Okay? All right. Any questions on that? Good? All right, then. I think we've covered everything there. Yeah? Canaliculi. The bile duct. Right? And that's going to drain. So then, finally, when we look at the biliary apparatus, when we look at the drainage now, going back to gross anatomical, which I wasn't careful about here in this drawing, but now you see it in this figure. When we go back to gross anatomical, we've got the right and left lobes of the liver draining into the right and left hepatic ducts. All right? Those coming together as the common hepatic duct. All right? And if you're not digesting food at the moment, then that bile will travel via the cystic duct and fill the gallbladder. And the gallbladder will remove a lot of the liquid and concentrate it there. Okay? If you are eating a meal and that meal has fat in it, then your digestive tract will release a hormone called Cholysystokinin that stimulates the smooth muscle in the wall of the gallbladder to contract. All right? And so it will begin to eject its contents. All right? And those will travel via the cystic duct into the common bile duct. All right? When you're eating, you're actually getting the flow of bile directly from the liver as well as from the gallbladder. All right? And that travels then via the common bile duct. And what did we say? Common bile duct joins the main pancreatic duct at the Hepatopancreatic ampulla, all right, also known as the ampulla of Vater, all right, and those will then drain through the major duodenal papilla, always also known as the sphincter of Oddi, all right, into the duodenum, all right. So that's our biliary system, okay. When people have gallstones, what happens? Often they'll go in and remove their gallbladder, right? Or they can do lithotripsy. They can try to break those up using high-frequency sound, all right? But they may go in and remove the gallbladder, all right? Have we impaired function in a severe way? No, you've still got bile coming directly from the liver. You just can't produce it that quickly, so you have to make some choices in life. No more donuts, okay? All right, no more greasy stuff, all right, because it becomes a little more challenging to break down, to emulsify the fat if the meal has a lot of fat in it, all right? But that's about the only limitation there, okay? All right, I think that's all I have to say on to wrap up our digestive system. Yes?
Gallstones were calcium. Or that?
Yeah. Yeah, so you have the precipitation of salts there, and little tiny crystals form, and they become a nucleation center for those salts to begin to, but they form little stones. Okay, all right. Okay. Done? Good?